The goal of this procedure is to precisely measure the effect that clostridial neurotoxins have on synaptic transmission in networked cultures of neurons derived from mouse embryonic stem cells. This is accomplished by first adapting embryonic stem cells to a feeder cell-free suspension culture and maintaining adapted cells in a pluripotent state. The second step is to differentiate suspension-adapted stem cells into neurons, or ESNs, using a variation of the 4-4 differentiation technique. Next, ESNs are treated with a series of media to promote maturation into synaptically active, networked neurons. The final step is to treat ESNs with clostridial neurotoxins or other neurotoxins and measure the effects on monosynaptic activity using whole-cell patch clamp electrophysiology. Ultimately, changes in the spontaneous production of monosynaptic activity are quantified from electrophysiological recordings, normalized to age and lot-matched control neurons, and compared among different conditions such as doses, times, or drug treatments.
The main advantage of this technique is that ESNs are the first cell-based model to replicate the functional pathologies responsible for the clinical manifestations of botulism and tetanus.
We first had the idea for this method when we found that ESNs were highly susceptible to all clostridial neurotoxins based on snare protein cleavage, and also demonstrated spontaneous synaptic activity and emergent network behavior.
Demonstrating the procedure will be Miss Megan Lyman, a technician from my laboratory.
Begin by transferring feeder cell-free suspension culture adapted ESC aggregates to a 15 ml conical